Hello, it's my pleasure to welcome you to this virtual scientific meeting developed by the American Neurogastroenterology and Motility Society, entitled What are the Basic Mechanisms Underlying the Effect of Diet on Symptoms of IBS? My name is Beverly Greenwood van Meerveld, and I'm the immediate past president of the ANMS and current director of the ANMS Institute. And before we start today's session, I'd like to remind attendees of a few important dates. The first is the abstract deadline for our annual meeting. This is March 8th, 2023, will be our abstract deadline. The date of the meeting is August 11th to 13th in Austin, Texas. Another date I'd like you to remember is the application deadline of December the 1st for the ANMS Basic Science Mini Sabbatical Program. And finally, I'd like to remind attendees about the ANMS workshop hosted by ANMS members at Stanford. It's a hands-on workshop, and it will be Saturday, February the 23rd, 2023. And it's now my great pleasure to introduce today's moderators. First, Dr. William Chey. Dr. Chey received his BA degree from the University of Pennsylvania, a medical degree in training in internal medicine at Emory University School of Medicine. He completed a fellowship in gastroenterology and has remained on the faculty at the University of Michigan in Ann Arbor, where he is currently the Timothy T. Nostran Collegial Professor of Gastroenterology. He's also chief of the Division of Gastroenterology and Hepatology, and he holds a joint appointment in the Department of Nutritional Sciences. Our second moderator is Bethany Doerfler. She's a registered dietitian, and she currently practices in the Division of Gastroenterology and Hepatology at Northwestern Medicine in Chicago, Illinois. She is the first dietitian to be fully integrated into a gastroenterology division for both research and patient care. Ms. Doerfler earned a bachelor's in science in nutrition from Michigan State University and a master's of science in nutritional epidemiology from the University of Illinois at Chicago. Ms. Doerfler's clinical practice and research focuses on providing wellness-based medical nutrition therapy for digestive disorders and allergic bowel disorders. And she not only presents her research to corporate, medical, and academic audiences, but her work has also been featured on the Today Show, Women's Health, Prevention Magazine, Chicago Tribune, Shape Magazine, and the Daily Burn. I'll now turn over today's event to our moderator, Dr. Chey. Thank you, Dr. Chey.
Thanks, Beverly, and thanks to the ANMS for inviting me to co-moderate along with Bethany this, I think what you'll find very interesting symposium, Basic Science or Basic Concepts in Neurogastroenterology, this webinar that'll be addressing the impact of diet on symptoms in IBS and really helping us to better understand the basic mechanisms that underlie those symptoms. So for our first talk, we're very pleased to welcome Dr. Boeckxstaens from Leuven, Belgium. Professor Boeckxstaens received his medical degree and a PhD degree and subsequent clinical training in internal medicine and gastroenterology at the Academic Medical Center in Amsterdam. He quickly ascended to the rank of professor and in 2007 actually accepted a full professorship in medicine at the University of Leuven in Belgium in the Department of Gastroenterology. Subsequent to moving to Leuven, he's received several national and international prizes and received the prestigious European Research Council Advanced Grant, both in 2014 and 2019. He's also an associate editor at Gut and has published over 300 peer-reviewed manuscripts and has an impressive H-index of 69. His research interests really focus on neurogastroenterology and neuroimmune interactions in the gastrointestinal tract. And he's going to talk to us today about local immune response to food antigen drives meal-related, meal-induced abdominal pain. So without further ado, Guy.
Thank you for the kind introduction. Let me share my screen first. I hope everyone can see my screen very well. And so, first of all, thanks for the kind introduction and the invitation to present today. I will try to give you some insights in the current new insight on local immune response to food antigens and its role in meal-induced abdominal pain. I think it's clear to all of you that stress and the gastrointestinal infection are the two main risk factors to develop irritable bowel syndrome. What is also mentioned quite frequently by patients, in approximately 84% of patients in this study, is that food intake is one of the main triggers to evoke symptoms in patients with irritable bowel syndrome. And it's of course important to understand what is the mechanism behind the induction of symptoms when patients ingest a meal. Of course, we all know about FODMAPs. Probably we'll hear more about this later. But what is really intriguing is the new insight that the mucosal immune response may be involved in triggering food-induced symptoms. And so the first evidence, in fact, that a food antigen can induce some kind of a reaction of the mucosa was provided by studies reported a few years ago already by the group of Fritsche Ravens in gastroenterology. What they did is they tested the effect of a perfusion of food antigens during endoscopy, during an upper endoscopy. And the food antigen that they studied was cow's milk, soy, wheat, and yeast. And they used confocal endomicroscopy, which allows you to visualize the response of the mucosa. And they could check secretion, for example, by infusing fluorescein IV, and as such see what happens if the mucosa is perfused with certain food antigens. What they noticed is that in 22 of 36 IBS patients, there was an immediate response to mucosal application of these food antigens. And here you see some images, and what you can see here is individual epithelial cells that really sort of secrete the fluorescent dye that has been injected. And in addition, what these investigators noticed was an increase in density of intraepithelial lymphocytes. So in other words, there appears to occur an immediate response when the mucosa is exposed to food antigens, at least in IBS patients, and not at all in healthy controls. This is really peculiar because our immune system is trained not to respond to food antigens, of course, and we refer to this as oral tolerance. This is a rather complicated process where macrophages sense the luminal antigens. They present it to the mesenteric lymph node, where regulatory T cells are instructed not to respond to a next exposure to this particular antigen. So we're trained not to respond to food antigens. That's the natural response if we ingest the food antigen. Of course, things can go wrong, and we can break this oral tolerance. And this is due to environmental triggers and happens in patients with a genetic susceptibility. And this is proven already in, for example, following a reovirus infection, to be responsible for the break of tolerance in patients with celiac disease and a following Th1 immune response. Now, we reasoned that perhaps this break in tolerance, or the reason why patients with IBS may respond to food antigens, could be due to a similar break in tolerance, but in this case, due to pathogenic bacteria. And the reason for thinking this is mainly because we know that an infectious gastroenteritis is an important risk factor to develop irritable bowel syndrome. I show you here a review by Giovanni Barbera, where he lists the different studies that reported on the risk of developing irritable bowel syndrome after an infection, either with bacteria, protozoa, or viruses. And you can see that this prevalence or this incidence changes from 4 to up to 54%, depending on the region and the bug that has been studied. So an infection can perhaps, at least that was our hypothesis, break this oral tolerance and lead to an immune response. So normally we have oral tolerance, and if we are re-exposed to that particular food antigen, no immune response occurs. But we figured that if someone is exposed to a food antigen, in the presence of an infection, this would trigger an adaptive immune response with the generation of antibodies that sensitized mast cells, and upon re-exposure of that same food antigen, these mast cells would degranulate and activate and sensitize afferent nerve endings, leading to abdominal pain. That was our hypothesis. And to prove this hypothesis, we developed a mouse model where we checked visceral pain by inflating a balloon in the colorectum. These mice were equipped with a transmitter connected with electrodes mounted or sutured to the abdominal wall. And so if we distend the balloon, the mouse has pain, it will contract its abdomen, and this then can be recorded by the EMG signal that is transmitted by this transmitter. So if we distend the colon with increasing volumes, we can sort of develop or make a distension response curve. We can calculate the area under the curve, and that is what you will see in the following slides as a proxy of pain in these mice. So mice were equipped with a transmitter. They were allowed to recover. We measured a basal EMG or a basal pain response, and then mice were infected with Campylobacter rodentium in the presence or in the absence of albumin, which we used as a marker food antigen. During infection, we recorded pain responses. Mice were allowed to recover. We measured pain once more, and then we started gavaging of albumin. So mice were re-exposed to the food antigen, and we checked whether an immune response had occurred and whether pain was altered when we performed colorectal distension. That was the protocol, and this is what we found. So 4 different groups, infected, non-infected, exposed to ovalbumin during the infection or not. And when we measure pain of these animals during the infection, so at 2 weeks, all groups that were infected, of course, develop increased abdominal pain, as you can see here, which is what we know. If a gut is inflamed, it will respond more extensively to distension. When we let mice recover and we repeated EMG recordings at 4 weeks, pain responses normalized in all groups. But when we started gavaging the mice and we measured pain responses, it's clear that there is only one group that has an extremely abnormal pain response, and that is the group that was infected and was exposed to the food antigen and was re-exposed a few weeks later with that particular food antigen. So meaning that pain response was altered and that most likely an immune response was evoked. Interesting, we only saw this in biopsy mice, of which we know that they have Th2 response, so some kind of an allergic phenotype, which was not the case in C57 black 6 mice, which are known to have a Th1, Th17 background. This could be relevant, as we know that patients with irritable bowel syndrome, especially if they have an atopic background, the risk to develop IBS is increased. And so this is somewhat in line with what we notice in clinical practice. The model is a sustained phenotype. So if we stop gavaging, pain remains. In other words, we are dealing with a model that is quite reproducible and produces a sustained phenotype. There is a lot to do about microscopic inflammation as a potential cause of irritable bowel syndrome. So we checked the presence of immune cell infiltrates. There were no increase in immune cells. And if we check for gene expression, the only gene that we noticed that was upregulated was the gene for encoding tryptase in the group that was visceral hypersensitive, and the group that was infected and exposed to ova, so sensitized. This group also showed an increase in gene expression of tryptase. Now we know that tryptase is a molecule, is an enzyme that is mainly produced by mast cells. So we figured that mast cells could be key players in the development of this visceral hypersensitivity. And so what we did is we checked the response of these animals, hypersensitive animals, to Boeckxstaens. And Boeckxstaens is a mast cell stabilizing compound. And so if we treat these mice after they developed visceral hypersensitivity with Boeckxstaens, you can see that the pain response normalizes while mice that were treated with saline or with vehicle, they remained hypersensitive. So the data that you see here is this area under the curve, and when it's above 4 and 4.5, we consider a mouse as hypersensitive. So you can see that pain responses were normalized in mice where a mast cell stabilizer was introduced. To further confirm the involvement of mast cells, we used mast cell mice that lack mast cells, so CPA3 cream mice, and you can see that littermates develop increased hypersensitivity, while mice that are lacking mast cells fail to develop this phenotype. So further confirming that mast cells are indeed important players in the development of this phenotype. Is this only happening when a mouse is infected, or so post-infectious? We also know that stress is an important trigger. And so if we stress mice and we expose these mice during the stressful episode to ovalbumin in their drinking water, in fact, we get a similar phenotype as if we infect. So if we stress mice and we then, after a few weeks, expose them again to ovalbumin, so we feed them ovalbumin, then we see that only mice that have developed or that were exposed to ovalbumin and to stress, if we start refeeding them from here onwards, you can see that only this group starts to develop increased abdominal pain perception on colorectal distension. So apparently also stress is capable of breaking this oral tolerance, and this is further investigated at the present time in the group of Stephen Vanner in collaboration with our group. Interestingly, similar as the post-infectious model, you can see that only tryptase, the gene encoding for tryptase, is upregulated in the stress in the visceral hypersensitive group. And again, if we stabilize mast cells, you can nicely appreciate that the phenotype disappears. In other words, that pain perception normalizes. Now, the key question clearly is what then activates these mast cells? And there's at least two main mechanisms or pathways by which mast cells can be activated. The best known clearly is IgE-mediated activation, which is the pathway that we know is involved in food allergy. Now, we first then checked, or at least one of the things that we checked first was to what extent IgE-mediated mast cell activation could be the mechanism involved in generating this phenotype. To do so, the first thing we did was to see if there was ova-specific circulating IgEs. This would mean that we had to do with a food allergy model if this was the case, but this was not the case. So in other words, we are not dealing with food allergy. What we noticed though is that if we measure ova-specific IgE antibodies in the colon, that we do see an increase in these ova-specific IgE antibodies, but only in the group of the visceral hypersensitive mice. So we're dealing with a local immune response, not with a systemic immune response. And to further prove that indeed IgE-mediated mast cell activation was involved, we treated these mice with a monoclonal IgE antibody, so to block and capture IgE antibodies. And if we do so, you can see that control mice develop hypersensitivity, while mice treated with this monoclonal IgE antibody fail to do so. In other words, IgE antibodies are indeed involved in activation of these mast cells. So this is what we end up then, that there is an adaptive immune response with the generation of ova-specific antibodies. We've proven that B cells and plasma cells are involved. I don't have time to go into this. And so the next question then was, what if mast cells are activated? What is the mechanism that then leads to this hypersensitivity, so this sensitization of these afferent pain-sensing nerve fibers? And this is work that was done in Kingston in the group of Stephen Vanner. So what they did is they took dorsal root ganglia. They were cultured and they were exposed to the supernatant of either normal sensitive or hypersensitive colons. And then the excitability of these neurons was checked during patch clamp, where two parameters were evaluated that are known to inform us on the excitability of the neurons. One is the rheobase, and the rheobase is the membrane potential at which the first action potential occurs. And the second parameter is the action potential numbers when the membrane potential of neurons is placed at twice rheobase. And so what they noticed is that if you incubate dorsal root ganglia with the supernatant of normal sensitive mice compared to hypersensitive mice, you can see that the rheobase is significantly lowered in the presence of hypersensitive supernatant. With other words, the neurons become more excitable. And this is also observed when neurons are studied at twice rheobase. You see that neurons treated with a hypersensitive supernatant are more firing than neurons that have been incubated with normal sensitive supernatant. Here you can see the numeric values. And if we then treat these neurons with an histamine 1 receptor antagonist, you can reverse this phenotype, and you can see that rheobase increases and that the number of action potentials decreases when treated with this histamine 1 receptor antagonist, meaning that histamine is one of the key players that is responsible for making these nerve endings and these neurons more responsive to a stimulus. And so this is then the pathway that we end up with. We have a bacterial infection, psychological stress. We also show that certain enterotoxins can do exactly the same, namely breaking tolerance, production of local antigen-specific IgEs with mast cell sensitization and activation, release of histamine, and sensitization of neurons. Is this relevant to patients? To some extent, we repeated the experiment of the Fritz-Ravens group, where we now injected antigens in the mucosa of patients. These patients were first checked whether they were allergic to the food antigens that we investigated. So they had a negative skin prick test and negative serum IgE levels for wheat, gluten, milk, and soy. We injected the food antigens and then checked 12 minutes later what happened in the mucosa. So here you see the injection. This is a tattoo to see where we injected. You see a nice vascular pattern. This is the injection. You can see a little blip. And so 12 minutes later, we go back and check. This is the tattoo again. And you can see with histamine, which is a positive control, you see a nice swelling, loss of vascular pattern. So this is a positive reaction. In case we inject gluten in a healthy volunteer, you can see the tattoo, the injection, bleeding, no response. But if we do the same in a patient with IBS, you see the tattoo, you can see the nice swelling and loss of vascular. So there is an immune response towards the food antigen. This is a summary of the experiments that we did. You can see the response to soy, wheat, gluten, milk, and almost all patients responded to at least 2 out of 4 food antigens that we tested, while healthy controls did not. And then we checked whether these mast cells in patients are indeed loaded with IgE antibodies. And as you can see here, this is staining for c-kit. So for mast cells, this is IgE. And you can see that the mast cells are closer to nerve endings and that the intensity of the fluorescence to IgE is more pronounced. So indicating that mast cells are indeed important in this process. So this is the scheme then eventually that we ended up with. And as you can see, in steady state, there is tolerance to dietary antigens. This can be broken by a bacterial infection, psychological stress, which leads to food antigen targeted IgE antibodies, which then are the reason why these mast cells will activate upon re-exposure and sensitize neurons. So in fact, what we propose is that we are dealing with some kind of a spectrum of IgE-mediated mast cell activation. We have food allergy at the very extreme, and IBS, where we have a local reaction compared to a systemic, where IBS is a local, less severe, or the least severe part of this spectrum, meaning that we have to deal with an IgE-mediated local response. So if this is true, then of course mast cells are important targets. Histamine and proteases are important targets for treatment. We tested histamine using an histamine 1 receptor antagonist. A small study, we just finished a 200 patients randomized trial, which we are currently analyzing. But here you can see the result of this small study, where you can see that the H1 receptor antagonist Avastin improves symptoms in 46% of patients compared to 13% when we use placebo. And if we stop, symptoms return. There's potential other drugs that can be used to stabilize mast cells. They are listed here. We tested an H1 receptor antagonist, but there's several other options. And there's a nice commentary by Dr. Rothenberg that you can read in New England if you are interested. So this is what I had to tell you. And of course, what remains is to thank all the people that really did the work, and especially the collaboration with Stephen Vanner in Queen's University. Many thanks for your attention.
Thank you very much for that excellent presentation. I'm thrilled to introduce our next speaker, who is Dr. Stephen Vanner. He is a professor of medicine and director of the Gastrointestinal Disease Research Unit at Queen's University in Kingston, Canada. This is a cohesive multidisciplinary group of both basic and clinical scientists. His major research interest relates to pain signaling in the intestine, and he has a basic and clinical research program examining pain signaling in irritable bowel syndrome and inflammatory bowel disease. His program focuses on peripheral signaling mechanisms, including microbe-host interactions, and involves neuroactive mediators such as histamine and proteases, and their signaling to TRP and voltage-gated ion channels. Currently, he is also editor-in-chief at the Journal of Neurogastroenterology and Motility. And a quick reminder before I turn it over to Dr. Vanner, we encourage our audience to please ask questions and to put them in the Q&A portion of the presentation window. After Dr. Vanner's presentation, we'll open it up to questions for both speakers. Dr. Vanner, thank you.
Thanks very much, Bethany, and thank you to the ANMS for this opportunity to talk about this important topic. So we're going to change gears a little bit, and I'm going to be focusing on experimental reproducibility and some work that was related to this that was conducted in our laboratory. So I don't have any disclosures related to this. So there's been increasing attention and concern about reproducibility of research in the past decade. And this is the results of a survey done a few years ago now of investigators worldwide. And you can see that over half the investigators themselves had concerns about reproducibility, and over another third thought there was at least some issue here. And when you delve into this survey a little bit more, you can see, if you look on the left-hand panel, have you failed to reproduce an experiment, that right across the disciplines, the respondents have had significant concerns about either being able to reproduce their own work or someone else's work. And if you go to the right-hand panel, when asked about what were some of the factors contributing to this, you can see that there were a number of different concerns raised. One was about selective reporting. Another was pressure to publish, low statistical power, not replicated enough in the original lab, insufficient oversight methods, poor experimental design, raw data, and possibly even the fraud in some cases, and insufficient peer review. This study caught my attention of an online anonymous survey of about 140 trainees in the US, and they were asked whether they felt pressure to publish on certain findings. And you can see that almost 20% said that they did feel some pressure. An even greater number felt pressure to support the mentor's hypothesis, even when data didn't support it. And an even greater number knew of mentors who required lab members to have a high impact publication before they could move on. It's also evident that if you publish a study, as shown in the left-hand panel, that reproduces a previous study, that you're twice as likely to get it published if you successfully reproduce results than if you're unsuccessful in reproducing the results. And the right-hand panel asks investigators, what are they doing in their own labs? And over a third said that they don't have any internal procedures addressing this issue of reproducibility. And a lesser proportion have some rigor within their labs to varying degrees. The most popular strategy was having different members reproduce experiments in the lab. This has caught the attention, not surprisingly, of the biotech industry and the pharmaceutical industry, and particularly in the cancer domain, where very promising preclinical trials were failing when they went to clinical trials. And this was really highlighted by this study by Amgen, a biotech firm in California, where they went back in the oncology field and found they could only reproduce 6 of 53 preclinical studies. And this was similarly found in a German study where they were only able to reproduce 25% of the studies. And as I mentioned, this now has caught the attention of the commercialization of drugs and investors. And you can see that this was highlighted in The Economist. But the bottom line is that if we can't replicate our science, then we're not going to be able to translate our science. Now, we've known about many of the different parameters. So sex and age, genetic background, litter mates, the different housing of the animals, diet composition, and sample size and biological replicates as just some of those. And today I'd like to focus a little bit more on diet composition and shine a light on this as just one of the important factors. Now, we know that dietary intake has a big impact on the microbiota, as well as either directly or indirectly on host disease. And some examples are shown here, and on biological effects and metabolism, immune responses within the body. In the clinical realm, in motility disorders, we've been particularly interested to date in FODMAPs and gluten. So FODMAPs, for those who are not familiar with them, are fermentable oligodimonosaccharides and polyols. And this is a fancy acronym for healthy foods that have complex carbohydrates that are not as well absorbed. And so they escape into the colon and the cecum, where they're then fermented by the bacteria, producing gas and also potentially neuroactive metabolites. And similarly, many patients who don't have celiac disease feel better when they take a gluten-free diet. So there's been a lot of interest in these particular diets, either a low FODMAP diet or a gluten-free diet in the clinical realm. And we are also, because of this, interested in mechanistic preclinical studies to try and understand and gain insights into these disorders by looking at the mechanism of action. So this led to us conducting a study that we reported a few years ago, and this was led by Caroline Tuck, who's a PhD dietitian who was a postdoc in my lab. And Caroline now is back in Melbourne, Australia, and has a very successful research program of her own at La Trobe University. So the aims of this study was to first gain some insight into commercially available mouse diets and just see what the variability out there was. And then we wanted to analyze nutrient content of these diets with a particular focus on FODMAPs and gluten. And then finally, to gain some insight into the potential impact of these diets on microbiome profiles and fermentation products. So the first thing that Caroline did was she did a Qualtrics survey on the listserv of the American Association of Laboratory Animal Science. And the response rate was not great, but I think it still provided us very important and interesting insights. So we heard from 39 laboratories in 7 countries, in North America, in Ireland, India, Australia, and the Caribbean. And what we found was there were at least 29 different diets that were being used. And this is a list of those diets and the relative response rates. So you can see that there's incredible variability in diets. And then there's also variability in what happens to those diets when they reach our animal care facilities. Some of them are irradiated and some are not, just as an example. So we wanted to look at the FODMAP content and the gluten content of these. And this was conducted in Peter Gibson's lab, the FODMAP analysis using their HPLC techniques to accurately measure this. And you can see there's quite a dramatic difference in the content. So over here with this particular diet, that there's a very low FODMAP content, and this is right at the level of if you arrange to receive a custom low FODMAP chow. And at the other extreme, you've got a diet that's very high in FODMAPs. Similarly, we found that there was great variability in the gluten content between these different diets. And just on average, there was a rough approximation that diets high in gluten tended to be high in FODMAPs. And this wasn't, I guess, particularly surprising because these both gluten and FODMAPs coexist in grains such as wheat. We also found that the water supply was quite different between various institutions. And it's something that I certainly had never really thought about before, but there's literature out there showing that the water supply alone can affect the microbiota within the gastrointestinal tract, and this in turn can have functional implications of how animals might respond, for example, in an inflammatory model. The final thing we found was marked differences in the grain, in the fiber that was in these differing diets. And obviously fiber is important when we're looking at transit time and motility disorders. So we then went on to study these diets, and Caroline set up the following paradigm. So we had 4 diets, as shown on the left, A, B, C, and D. And the first diet, the animals were C56 black 7 mice from Charles River in Canada. And these animals were housed here at our institution for a week, and then tissue was harvested. And then the other 3 diets were either moderate FODMAP gluten, low FODMAP gluten, or high FODMAP gluten. And what we found was, this is the 16S RNA sequencing data. And when we do this NMDS plots, we can see that the microbiota profiles separate out based on the diet. And if you look a little more carefully at the circles and the triangles, you can see they also separate out on the sex of the mice. We went on to look at the genus level, and you can see here across the top, you've got diet A, B, C, and D, and male and female. And the black boxes are significant associations with the diet. And you can see again that at the genus level, there's significant differences in the different diets on the expression of the bacteria. To give some measure of functional implications, we took the cecal contents and measured short-chain fatty acids shown in panel A, branched-chain fatty acids shown in panel B, and pH as shown in panel C. And you can see that there are significant differences. For example, in panel A, the low FODMAP diet C had a significant reduction in short-chain fatty acids. There was a significant increase with this diet in branched-chain fatty acids. And here with diet B, which is the low FODMAP diet, not surprisingly, the pH was significantly lower because there was, and in this diet with the higher FODMAP, significantly higher because of differences in fermentation. Now, these obviously are indirect measures of functional responses, but there are several studies out there looking at different diets that have been shown in preclinical models that the diet can directly impact, for example, the degree of inflammation in a model such as DSS colitis, for example. So in summary, these commercially rodent diets significantly differ in their nutrient content, and these can have a significant effect on the gut microbiota, community structure, and on functional outputs such as pH and short-chain fatty acid profiles. So we think that the rodent diet, and there's ample evidence in the literature for this, can underlie poor reproducibility, and particularly of concern in studies looking at gut microbiota. So I think the takeaway here is that we need to pay greater attention to diet, and we need more detailed reporting of preclinical studies, especially those looking at the microbiota. And it is interesting that journals such as Nature have taken away the restriction on the length of the methods section in their papers, specific to address concerns such as this. I just want to end by mentioning that for those of us who do clinical trials, it is interesting that there's much more rigor related to these. So we have institutional oversight. You can't conduct a clinical trial without having completed good clinical practice training. We've got institutional oversight on the quality of the research that's being conducted, and we've got regulatory oversight that not infrequently visit the institution and make sure that good clinical practice guidelines are being followed, and they're quite rigorous, and that, for example, all the equipment's being calibrated on a regular basis. And this, of course, is not happening to the same degree in the preclinical studies. But there are guidelines out there, and I just wanted to draw your attention to these. So these are the ARRIVE Essential 10 guidelines that can be applied to preclinical studies. This is a checklist from Nature for editors and grant reviewers for rigor in preclinical and clinical studies. And then finally, this is a paper in AJP in 2018 with recommendations for microbiota researchers that evolved out of a NIH workshop. And I'd encourage you to have a look at these different guidelines. So I'll end there, and thank you very much for your attention.
Thanks, Steve. So just wanted to remind everybody, please send in your questions if you have any about both of these provocative and very interesting talks. Steve, that's, I have to admit, I'm not a basic scientist, but I hadn't ever thought about that to the degree of depth that you've gone into, and it's mind-boggling to actually think about the implications of what you just talked about. I mean, you really do. Are there any discussions about developing an international consensus just to help guide people that are doing the types of research that you and Guy are doing? Because it strikes me that it's profound what you just talked about.
So I agree, Bill, and so I'm not aware of any talks, but I mean, it's certainly going on. I know at, for example, NIH grant review panels, and so this kind of rigor is now being applied. So I think it's certainly been humbling for me actually to go back and sort of walk through this literature, and it's going to change what we do here locally in our lab for sure. And I've already been talking to our students about it. So it's been helpful for me to prepare this presentation.
Yeah, wow.
Mm-hmm. Yeah.
Bethany, you want to handle that one question for Guy?
Yeah, I mean, and also just interesting commentary on that, the last slide that you presented really strikes to the variability in animal studies diets and also human diets. The recommendation there is controlled feeding studies are ideal when assessing the microbiome in adults. And it really makes you appreciate some of the variability in findings, and that just human variability in diet in animals, that all of that and the water supply really can add to things. So I really appreciated learning that. To ask this particular question from Dr. Parkman, for Guy, what H1 agonists are used clinically for IBS? And is this really just for IBS with diarrhea?
I'm using H1 antagonists, so not agonists. I'm using an H1 antagonist. We are using Avastin. That's the one that we have been using in these trials. There are some patients that are on H1 antagonists for their allergic rhinitis, but I preferably switch to the one that we have experience with. But in fact, I think you can use any H1 antagonist available. What is important, though, is that we use double dose and not single dose. The study that I presented was single dose, but we have experience now that double dose is much more effective. And what is the most important remark that I want to make is that you need to treat long enough. So if you need to treat for 6 to 8 weeks at least, that's when patients start reporting improvement. We don't have a clear explanation yet, other than that we think that the phenotype of the neurons needs to be readjusted. But that's a major error made so far by GPs and gastroenterologists that are starting to use this treatment here in Belgium. So treat long enough and take double dose. That's really important. We not only use it for IBSD, I also use it for mixed and non-constipated IBS. But some patients with IBS-C are also treated and helped by this treatment. It's, of course, the small study that we did, as I mentioned, is about 50% of patients that respond. I hope to have more info on success rates and other criteria when the current larger trial is finished.
You know, Guy, I follow on to that. Some of us are old enough to remember that, you know, our pediatric GI colleagues for years and years, decades and decades actually, have been using antihistamines to treat so-called functional abdominal pain or recurrent abdominal pain, what we used to call as RAP. And you know, periactin is one of their most popular drugs, ciproheptadine, which you will remember actually decades ago there were studies with ciproheptadine treating IBS that suggested benefit. So in some ways, it's kind of back to the future. It's a very interesting time. And following on that, what about mast cell stabilizers? You know, I mean, remember back in the 90s, there were also studies using cromolyn sodium for patients with IBS and reporting some positive results. Is that something that clinicians can think about as well?
Of course. The problem is that we have no efficient mast cell stabilizers at the moment. And those that are a bit stabilizing mast cells have many side effects, like patients get very sleepy and dizzy of these compounds. But for sure, mast cell stabilization is I think, an important new trajectory to further investigate. There is a few case reports on omalizumab. And this is interesting because that's a monoclonal IgE antibody used for therapy-resistant asthma and eczema. Mm-hmm. And there is case reports with patients that were treated with this and concomitant IBS that they improved. Of course, this is a very expensive treatment, but it proves that the concept of stabilizing mast cells could be an interesting one, and it's definitely something to be further explored.
You know, Guy, I also wanted to ask you, Bethany, is it okay if I ask one more question?
Please, yes.
You know, there's yesterday in the United States, and I don't know if this was also true in Leuven, but there was a big news story that broke about genetic markers identifying patients that were less likely to die from the bubonic plague. So in other words, they unearthed bones and artifacts that they could derive DNA from folks around the time of the bubonic plague. And what they found was that there were four SNPs that identified patients that had a survival advantage relative to individuals that don't have these SNPs. And what's even more interesting about that, and to build on your work, I just want to get your comment about that. I don't know if you saw this article, but what they found was that those individuals that had the survival, that had these SNPs and the survival advantage, their descendants had a dramatically increased risk of developing autoimmune diseases like Crohn's disease. So there, sort of top line hypothesis is that there are individuals that have mutations that confer a survival advantage for infections, but this hyperimmune immune system may predispose to developing, autoimmune phenomenon. And it's just, as I was listening to your talk, it sort of brought me to that discussion, because it's really a fascinating new way to think about these conditions like IBS. And the question I have for you is, what do we know about epigenetics, in regards to some of the findings that you described?
Not much, really.
Yeah.
But I think our genetic makeup is really important. Another example is, for example, achalasia. Why do certain people, certain patients, sort of attack their own neurons to keep in the neurogastro field? Possibly triggered by a viral infection, at least that's the current hypothesis. So why is that? Also probably because there is some kind of an immunogenetic background. And as I mentioned, for patients with irritable bowel syndrome, there is an increased risk if you have an atopic background, which is compatible with a Th2 background, which we also see in our model. So BALB/c mice developed a phenotype. Black 6 mice, which have a completely different immunogenetic background, do not. So I'm pretty convinced that we should pay more attention on our immunogenetic background to understand why certain people develop a disease and others don't, and how to search for this, how to prove this. Yeah, that's another story, of course.
Yeah, Steve, it looks like you have a comment.
Thanks. I have a follow-on question actually related to the points that you both are making, and I just wanted to ask Guy, do you foresee us finding biomarkers that might predict the beneficial treatment for somebody for the mechanism that you're talking about, whether it's on the immune side or something in the mucosa?
Well, actually, a little bit based on work that you've been doing as well, Stephen, is if mast cells are an important player in the pathophysiology, looking for mediators released by these cells, either in feces or urine or in biopsies, but of course, the least invasive we are, the better, could be of help. So what we are currently doing, and I'm pretty sure you're doing that as well, is that we are looking for histamine metabolites in urine and in feces, and to see if we can link this to the therapeutic response in the patients that we have included in our Avastin trial that we are currently analyzing. So if we can see that we can predict based on urine histamine levels or fecal samples, then we can start stratifying patients and improve the outcome of our clinical trials if we want to use mast cell stabilizers. And I hope that this will be very important for pharmaceutical companies to get back interested in supporting clinical trials in the IBS field. So I think it's an important point that you make, Stephen, and this is really important for the field to further develop and get interest of pharmaceutical companies again.
Do we have time for one final question, or should we wrap it up?
I think we're out of time.
We're out of time. Yeah, we're right on the hour. We would love to thank everyone for attending today. This was an outstanding discussion, really novel research presented on the part of both of the speakers, and we thank you for presenting and teaching us today. Thank you to the ANMS and to my colleague, Dr. Chey, for participating today. There are no additional questions in the Q&A, but if any arise, they can be answered on DocMatter. And we also would like to highlight this URL as a place for discussion, for burning clinical matters, and for recordings of this and other videos. Thank you for your time today and your attendance. Have a great day.